I'm Craig Beasley from Biogen, and I'm going to talk about what do we think is our next generation manufacturing. People ask me all the time, what is next generation manufacturing? I think the real answer is it's what you need at the time. And if you look at Biogen's current manufacturing network, we have similar areas. We started with 2,000-liter facilities, and now we have 8 2,000-liter trains spread between Cambridge and North Carolina. Then we built 15,000-liter reactors in North Carolina. And I actually spent 4 years in Denmark building our next 6 15,000-liter reactors, and I was told they're dinosaurs, they'll never be needed. You might as well mothball them at the time. So we went ahead and built the flexible volume manufacturing in North Carolina. And that's been fantastic. That's really helped us get first to patients, first in humans, and phase 1 clinical product into the trials very efficiently. So that's, at that time, that was our next generation. And it's been transformational, and it's really shortened the clinical time. But now we have a very specific target commercial patients. We're in multiple sclerosis and we're in hemophilia. So roughly we serve around 210,000 patients, and we're very proud of that. I think over the years we've had a great compliance record. We've valued getting patients' products wherever, whenever, and we've done a good job. And we want to continue that. I think that's what we owe our patients, we owe our shareholders, and we owe our employees. So how do we do that now when we move to Alzheimer's? And Alzheimer's is obviously millions of patients, and unfortunately, I'm sure many of us have been touched by that in our lives. So how do we take the best in what Biogen does and then replicate it in a way that we can still serve our patients without building 4 new facilities of 15,000 liters? And that's where we questioned. We think we have a product that will be ready in 2019 and 2020. Now, we still need some good luck and some good hope and some good science for that. But we can't wait until the clinical data comes out before we build our facilities. So our solution was to build a manufacturing plant that really could produce 10 metric tons. And we chose to build that facility in Switzerland. So what I'm going to do today is tell you a little bit about the facility, but mostly how we're going to operate the facility. And just, this is the picture 4 or 5 weeks ago. We broke ground in January. We started putting up steel in April. My engineers would kill me if I didn't say we're putting up 800 tons of steel a week. So we're making some good progress. The view behind the plant really does help the plant, I'm sure. So that's quite nice. But the real goal is to make sure that we are 3 to 5 times more productive than the current facility. And we're going to use what we call 3X technology, which I'll touch on a little bit after this. And then we're getting back to basics. There's many industries, and when we say next generation, we're saying next generation to Biogen, next generation to lots of biotech. There's industry out there that have 6 Sigma quality every time. You don't want to get on a plane unless it's up to that. And biotech isn't known for that, right? We brag about 98, 99% success rates. We're here to argue that the process development data is here, the analytics is here, that we can start approaching 6 Sigma, and we should approach 6 Sigma level of quality. So we'll get back to basics, do some process mapping, and really put lean into our manufacturing process. And again, we don't want to build a facility that sits idle. Many people have been through that. It's not so fun. I've been in selling my plants before. I don't want to do that again. So we've designed this facility to be modular. We'll build out 2 cells. And we're ruthless in our engineering that we will then copy identically to build cells 3, 4, and 5. Because we think the patients don't want to pay for your innovation to go from cell 2 to cell 3 and make sure that we get a little bit better. We need to develop the processes that make us better. The clinical and the product itself needs to be better. But once we have a workable design, we'll replicate that design. And then we will then implement full recipe operation. But what we want to do with our high level of automation is make sure that we can actually service both clinical and commercial. And I'll touch a little bit about that because we don't think that you have to lose flexibility by being highly automated. There's some, but it doesn't make sense to pay 10 automation engineers when you could just pay 10 operators, right? So you have to be smart about how you do automation. And we're going to try to approach that. And we're going to use all of this to better understand our process and make sure that we build our facility by 2019, have it at least licensure ready. Now we still have to execute in the clinical trial so that we obtain that. And we use advanced process control, or at least what we call advanced process control, to make sure that we don't start up at 84, 85% success rate. We're going to start up with the right time release and do it right the first time. That's our approach. Now, what we actually believe in Biogen is Oslo, we're a partner to the community, and we're a partner to the environment. So if we do these things with this efficiency, I'm going to show you a little bit of data that we will make a much less impact on the environment at the same time. So our basis of design is actually fairly straightforward. We'll initially build 2 manufacturing cells. What's in the cells? 4 18,000-liter reactors with 1 purification suite. Each cell is designed, at least from utilities and process equipment, to go between 5 grams to 15 grams per liter. And right now we're targeting between 10 and 11 grams. And we think we can do that with some of our processes. Not all, but some of our processes. And then we'll do a very typical design within the biotech world, have a central core of the utilities, so that we can expand very quickly without disrupting ongoing operations. And I'll walk through a little bit. This is the site master plan. Again, nothing novel from the outside. We'll have a central utility building that you'll trust me with. Central utility building, 2 manufacturing cells, and then as we grow, we can expand the utility buildings and expand the manufacturing cells and warehouses needed. What you'll not notice is there's not a big lab building, and there's not a big administration building, and we'll talk a little bit about that shortly. We started a design with some guiding principles. Use science to drive decisions, use technology to enable the systems, and work across boundaries. What do we mean by that? We wanted to build our facility in a way that really focused on the entire team of the drug substance manufacturing process. I know when you first go into a manufacturing area, you get the org chart and it says manufacturing, process development, quality, engineering. Our goal is when you walk into our facility, there's one drug substance team. Different responsibilities, different accountability, but when you look at the team, you should see one team. And we tried to design that into our system to force that. All the information about the products and the processes need to be available, and this goes from end to end, from clinical all the way to commercial, so that when you have an investigation or you need that data, it's at people's fingertips. They don't have to walk into an admin building. It's available at the manufacturing floor. And that should allow decision-making about the process and the product to be immediate and at the point where decisions actually make impact, which is on the manufacturing floor. With all that being said, especially when you go into older facilities, the manufacturing floor isn't always the best place. There's some places worse than others, as we've heard. But even we've designed facilities where the light's not in, there's no natural lighting. It's kind of cramped. We're trying to define a facility that actually the manufacturing floor is where people want to be. And we think with closed systems, now you can operate more things in CNC spaces. You're able to replicate the comfortable environment that people are academically engaged and excited to be there. So we're trying to do that. With that being said, there's no need to have a QC lab standing off. We're going to integrate our QC labs inside the manufacturing building, and then no reason to have big admin buildings. Put that in the manufacturing so everyone is there for one reason, which is drug substance manufacturing. So the design of the facility itself, even though I'm very proud of it and I think we've made some pretty cool designs, isn't how we think we're going to get the productivity. We think our new way of working is focused on 3 things. One is the manufacturing process itself. We call it 3X. Second is once you develop and produce a lot of protein, make sure you produce a lot of the right protein at the right quality, and that means advanced process control. And then have really smart, engaged people that can actually operate it. But you need to do that in places unlike Cambridge. Cambridge, there's a lot of people that are skilled, qualified. We're building in Solothurn, Switzerland, which is actually a very great place, but it's not the biotech center of the world, right? They're good at manufacturing in Bosch and other tools, but we have to make it in a place that we can actually hire local people and replicate it in the way we want to do it. So for the cell culture, we do have higher cell lines. They've been optimized so that we can produce the 10 grams per liter. We do get the efficiency of perfusion reactors, and I'll walk you through a little bit, and then we've had a lot of problems of how do you get that through purification, and John gave a talk earlier this week to kind of go through details of how we're tackling that purification problem. So I won't go into too much details for that. But for process control, look, I think all of us have been in the place where in 2006 or 2007, we knew how to measure many of our processes, but we couldn't change everything for the same reasons you talked about earlier. Is if you changed one process, then you would have to wait to change the other, and you have to file in the 79 different markets around the world. And by the time you got it approved, you would stock out a product. So we feel like now's the time. We have a fresh start for a manufacturing facility, a new product, the technology's there, the vendors are excited, and the community is excited. So we're gonna leverage all these things that we wanted to do a while ago and put it in place. And the technology has caught up a little bit, so we'll talk a little bit about that. But then you build in and really fight the paradigm that the way to make good manufacturing product is to do it the same way every time. And we actually think you do want to do it the same way every time, but as you know, there's inputs that change. So we need to design our process and the controls that actually changes how we move a biologics product. So you might change the number of column cycles you run to actually make sure the product is the same. And then many people have used predictive modeling to actually monitor the process. For multivariable analysis, we know that things interact. But we want to actually use that and actually push it a step further and use that for some batch disposition activities and really start working with the regulatory agencies to understand that, work with process development to really characterize that, and then make sure that we actually can do things efficiently. And then build all that in, we should then be able to use really smart people to run a really efficient operation, and we'll walk through that. So the improved process, people have used high-density cell banks for a while. That really reduces the time that it takes to get up to your bioreactors. We will implant perfusion reactors in the N-1 step, and that allows us to get the cell density and the cell mass to the point that we can always inoculate our production reactors at a high density. That also cuts down quite a bit of time, right? If you're not doing expanding cells, by doing that, really allows you to be much nimbler in your supply chain. I would actually argue the worst thing that you can do for a supply chain, but what we're forced to do, is build large inventories. And you do that for several reasons, right? Every time you need 6 batches, you make 7 because you need that one in progress so your facility is not holding up. And you have that backup, and then hopefully that goes to your safety stock. But when you actually compress it, you can be much more nimble. You don't have to have so many batches going on concurrently, so you're not wasting operation time. We have the cell productivity that we talked about, and then again, from the chromatography steps, we have to get that amount of material through it, and then use single pass ultrafiltration to actually reduce the volume that you need to do. So that allows us to make a whole lot of protein. We're excited about that. But that doesn't help if you can't make the right protein. So we know the biggest thing, most investigations are raw materials. The raw materials changes, you start your clinical, you only do 5 or 6 clinical batches before you get into phase 3. So we're taking advantage to work with our process development, to work with the suppliers of our raw materials and truly get a good characterization of the raw materials impact, and then change our process, design our process control so that we can limit that impact on the process. To do that, we need to actually understand what the raw materials impact. I'll get to that in a little bit. And then as the vendors actually get to know their raw materials much better, and as we get to know what we need in the raw materials, that allows us to take a lot of the weighted tests for raw material testing and just do an ID. So we'll leverage the supplier's raw material testing to do release of the raw materials. Process control, again, we're implementing at-line or online probe as much as possible, and we'll get into that. And then we're actually coming up with adaptive process controls in the purification process to change how we run our purification. With all of this information, then it allows us to do product testing, and we call it right time released. And what do we mean by right time released? It doesn't add us a whole lot of value to be able to release product the day after the manufacturing process is over. What it does is, if it's within 3 or 4 days, that's fantastic. But you don't want to carry a process past the first column if it's not going to pass. You want to get that testing done at the point when your critical process parameter changes last. Test it at the end of the first column or the second column if that's where the process is locked and there's no other levers for it to go through. And that way, by the time you actually have your drug substance, that test is done, released, and you haven't put any other batches across that process, unit process. And then obviously use all this data for modeling so that not only do we continually evolve and evaluate our manufacturing processes, but we can then use that for batch disposition or investigations. So what does that look like? Our advanced process control strategy is really to eliminate all batch failures, really start to increase our operational efficiencies to meet our patient supply and patient needs. Again, we screen raw materials, and then when we look at the production reactor, we use online probes, Raman technology to measure for glucose. We actually use it to measure for other parameters as well. And then we feed, like many people do pH decades ago, as you need. So if you have more cells, you feed more product. It doesn't sound novel, but it definitely limits the amount that you're impacting your cell culture, so you get a much more consistent process. You do the same thing with glucose, with nutrients, and you keep a really tight environment so that every cell is exposed to the same environment all the time. And then our process in the purification is really know your purification process well, know what variables change during the unit process, measure it before if you need to change it. So if we have high aggregate, we aren't just going to throw away that batch. We know that if we run more column steps, then we can actually reduce that. You'll also lose some yield, so you don't want to do that every time. But file your process so that in normal batches, you can do 3 columns or 3 cycles. If you need to remove a little bit of aggregate, split it up and use 4 cycles. So that allows us to hit these higher level batches with these one-off batches not being thrown away, but actually pulled back into process. Now it requires us to spend a lot more time in process development. And how do we counteract that? We actually just designed the process development work to accommodate that. So we believe we get a much higher consistent level of product quality. And that's great, but now you need to make that sustainable. So we are tackling that by 3 steps. One is a highly automated integrated system, full recipe, define how you want your process ran. Don't focus on a manufacturing execution system that looks top down, but focus on the unit modules themselves and let the process controllers actually drive their batch. Most of the time when you run a bioreactor step, it's not very different from one process to another. And when you run a column, it's not different from one process to another. What's difficult is when you add them up and you change the pieces. The typical manufacturing execution strategy focuses on a recipe that says you will do this, and the automation engineers spend a lot of time then remapping things. If you actually build it just like you would an SOP and you have libraries, and the libraries actually call for what samples to pull or what release tests need to be done, then it's much more modular, and you can switch between your clinical platform and your commercial platform. By putting this in place, then you really do get this end-to-end visibility. And what do I mean by that? When you're running your clinical batches for your phase 3, you have the data. When you're running it 3 years later for your commercial, you have all the data in unit operations, and you can actually compare them against each other to trend things to see if they're changing or to see if you need to change your manufacturing process. Our goal really is to also lead to execution systems that are based off of industry standards. I think we do, as an industry, we don't work as well together as we could. So we spend a lot of our time, and there's been a lot of manufacturing plants that are either don't start up on time or worse because the automation's not ready. That's not what makes us special as our automation. So we want to start building standards, sharing it between companies, and actually letting the process actually be the success story. If we do those things, we've also then designed the facility, as I mentioned briefly before, so the decisions are made on the floor. This is just a small layout. But within this room, we have a QC lab that's not only for the work in progress or the manufacturing process, but we actually put the release testing at the manufacturing floor. We build a nice environment so that we get sun in, and we build workstations so that people can work there, engineers, manufacturing, and quality. That's an integrated area, and as we start to see, in other industries have seen this as well, when you run manufacturing, you notice people start scurrying when they're about to pull samples. That's a visual cue that quality can get ready and start prepping the lab, so it's efficient. And then right time release. As we do all of these things, we've localized the labs. Samples aren't passing around. You're not spending carts walking from one facility to the other. They're moving from one room to the other room. Samples don't get lost. So we're trying to localize the sampling and really implement this samples never rest process. If you do all these things, then you can actually do exception released batch disposition. And then that data is available for if you need to do updates for regulatory filing. There's not this 3-month data collection process. It's there and available. So that's our approach. The nice thing about this, if you do these things and you do highly efficient processing, we're actually making a more sustainable manufacturing plant as well. At least our calculations today, and I'll verify it in 2019, we expect to have an 83% fewer carbon emissions, use roughly 80% less energy, and use roughly 90% less water. And that's a huge difference that we're very proud of. We're putting in solar panels so that we actually, anything that we cannot make neutral, we will use solar to create the difference so that non-process energy is at least neutral for us. And then again, implementing ergonomic design throughout the facility. So that's our approach. I think if we do those things, we'll set a standard that really allows us to meet our patient needs. And our goal for our next generation is really simple. For the Solotherm facility, we want to be able to go from start of January 2016 with the greenfield to by 2020 have 1 million patients receiving meaningful therapeutic products. To do that, we've developed a very simple project mission. It's every day counts. Every day we make an impact. Every day we go home healthy. And every day we transform not only our lives, but our patients' lives. So our commitment to our patients is we really do act every day that 1 million patients are counting on us every day, because they are. So that's our facility design.